Two computer programs developed from the Science and Technology series are available on disc for the BBC B Computer, together with a booklet for teachers. These can be obtained from GSN Educational Software, 214 Stamford Street, Ashton-under-Lyme, Lancashire, or from the Education Officer of your local ITV company.
A pint and a ploughman's, please. Technology uses scientific ideas in industry, perhaps to make chemicals or to produce and shape metals. Biotechnology makes use of living materials, microbes, either to change other things or because the microbes themselves are useful. Thank you.
That's 122, sir.
The use of living materials isn't new. Almost everything in this lunch was produced using microbes. Bacteria help to make the butter and the cheese from milk, and yeast help to shape the bread and to brew the beer. This small brewery produces beer in a way unchanged for centuries. Yeast is added to a mash of barley, sugar, and hops. With a food such as sugar, yeast cells grow in size and reproduce. Yeast changes the sugar to produce alcohol and carbon dioxide. When microbes like yeast grow and make useful substances, the reaction is called fermentation. And there's a vast range of such reactions, each using different microbes, different foodstuffs, and different growing conditions. Microbes themselves are stored in small quantities at very low temperatures, around minus 150 degrees Celsius. They start growth in a flask that contains food for the microbes. This transfer from store to flask needs to be made carefully to prevent contamination. The flask is kept at the best temperature for microbe growth. Shaking ensures a supply of oxygen. As the number of microbes increases, they are moved from the shake flask to a larger fermenter. The fermenter is fed with a continuous supply of oxygen. Foodstuff is fed in. And as the fermentation continues, microbes are continuously pumped out. The microbe will grow best at a certain temperature, and when stirred, at a particular rate. The growth will be most rapid within a narrow range of pH. This fermenter is laboratory size. In industry, the scale of the reaction can be vastly increased. Microbes can be produced by the ton. This is just the top of a large industrial fermenter. Microbes are being fed into the fermenter from the steel bottle. In some fermentations, biotechnologists are interested in the microbe itself. But here, the important byproduct is an antibiotic. The antibiotic is extracted as a powder. Technicians working on antibiotic production sometimes need to wear suits like these. The suit completely covers the body to prevent any chance of contamination. And each suit has one of these. An external air supply for breathing. Antibiotics are important as medicines because they can destroy harmful bacteria. One of the first, and still a most important use of fermentation, is in the production of such medicines. Washing powder works with the water in the washing machine to dislodge small particles of dirt from the clothing and to make sure that the same dirt doesn't get back onto the material. But there's a problem, and it's nothing to do with the washing powder itself. Many of the clothes we wear are made from synthetic fibres or from a mixture of perhaps cotton and synthetics, and often the material has been dyed. Hot water around 95 degrees Celsius will damage those fibres, and many dyes aren't fast at that high temperature. This label recommends that this shirt shouldn't be washed in water hotter than 40 degrees Celsius. The problem is that traditional washing powders don't work so efficiently at those low temperatures, and that's why you'll find two types of powder on the supermarket shelves. An original type, and next to it, a packet that will say biological. So, Malcolm, tell me what sort of marks washing powders have to remove.
Well, Max, I've got some examples here which show different sorts of stains and different degrees of difficulty.
Right, what's this one?
The first one's the most simple, really, and that's just mud or soil, which can be removed very easily once the detergent has lowered the surface tension of the water, and it can then just lift off the stain.
Right, and what's this one over here?
The next one is also very commonly found, and that's coloured marks caused by fruit juice, tea, coffee. Well, all powders contain a mild bleach, and that works by liberating oxygen in the wash liquor, and that just bleaches out the stain. A bit more difficult now, we come on to what we call proteinaceous stains.
Yeah.
Things like food stains, you can see on the tea towel here, egg and tomato ketchup. Body stains like grime on collars and cuffs. These contain protein, and the protein acts like a glue to stick the stain onto the fabric so that the detergent can't lift it away.
So, what's the answer?
The answer is to use a biological washing powder.
How does that work?
And that works because biological washing powders contain enzymes which have been grown in cell cultures, extracted and added in small amounts. And the enzymes react with the protein, break it up into small pieces so that the detergent can lift the stain away.
Washing powders are complicated mixtures, with each ingredient having a particular job. But this mixture doesn't happen by chance. It may follow years of testing to get it right. Technologists will test their new powders with load after load of dirty washing. Often, the washing machine's own programmer is bypassed and replaced by a computer. The computer ensures that the washing time is accurately controlled. There will be precise records of the amounts of water used for washing. Any differences in washing will then be because of differences in washing powder. These tea towels were washed in a traditional powder on the left and biological on the right. Technologists also wash test cloths that have known stains. This is a cocoa stain, before and after washing. And the wash water can be changed to represent different degrees of water hardness. These are blood stains, before and after washing. But not all tests of washing powder take place in the laboratory.
What I'm going to do now is I'm just going to wait the washing to see exactly how much you're going to put in. Make 3/4. And what I'd like you to do, on the blue towel that you've got there, if you will pin the test cloth.
Although washing powders may work well in computer-controlled machines, eventually those powders will need to work equally well at home.
Okay, so if you put it in the machine as you normally would do. What program are you going to use on this one? Number 4. Number 4. Okay, so if you actually would put the powder in for me now.
Different people will have different ideas about how to get the best from their machine.
2 of those. 2. Right.
Washing powders will need to do their job in a range of conditions. They need to work even when the amount of washing, or the quantity of powder, or the wash time, or temperature are not precisely what they should be. The coloured test materials are checked after each washing using a colour analyzer connected to a computer. The computer records the loss of colour, if any, from wash test to wash test.
Hey.
Some articles can just be compared with each other. Which white towel is the whitest?
A.
Biological washing powders were first developed some 20 years ago. And biological powders were different because they contained enzymes extracted from cell cultures to help solve a problem. If you wanted to grow plants, flowers or vegetables, then most of us would pop along to the local garden centre and buy a packet of seeds. Or perhaps if you were very skilled, you could collect cuttings from plants already in the garden. Biotechnologists have developed ways of growing plants in laboratory conditions. These plants will grow at any time of the year and can be cultivated in their thousands. The technique uses lengths of stem taken from a parent plant. The flower, together with the side shoots and leaves, are discarded. Each piece of this stem carries a bud. Some plants are easily damaged by microbes. Bleach is used to sterilize the small lengths of plant. Sterile equipment is used to transfer the plant stem to a growing medium, plant food. Sterile air blows round the containers to keep these delicate tissues microbe-free. The plants are kept warm and in artificial daylight. After 3 or 4 weeks in the growth room, plants are big enough to split into smaller pieces. Eventually, there may be tens of thousands of tiny plants. The growing medium is altered to encourage roots to grow, and the plant goes to the greenhouse.
The plants, when they're cultured in the growth room, are kept at about 100% relative humidity, and this has to be maintained on the nursery when they're planted out, otherwise they'd wilt and die. This is done by a fog system, which is high-pressure water droplets.
All these plants came from one parent. All the new plants will have the properties of that parent, and they'll be as strong and as disease-resistant as the parent plant.